Hi everyone, welcome back. Now we're going to be talking about immunizations. So at this point in the class, we've covered a lot of information about a diversity of microorganisms, a lot of which are pathogens. We've talked about bacteria at length. More recently, we explored viruses and the way that they work. And later on, we're going to be talking about the way that our body deals with these bacteria and viruses and other pathogens when we encounter them, how we fight off this infection when we get them. And after we have a successful battle with a pathogen, our immune system, it's educated. It gets experience from this fight that it just had. And it used that experience to build things like antibodies and create a much more targeted attack that allowed us to kind of look for a very specific thing, this very specific disease-causing organism inside of our body, fight it, kill it, and that experience, it doesn't just go away. It sticks around, and that is what we call immunity. When our immune system has immunity, it remembers this past infection, all these infections that it had before. It remembers how to fight them, which then becomes very useful if we ever encounter them again. It's often the case that we get exposed again and again to things that we have fought off in the past, but a lot of the time we don't even realize that we ever had. Because our immune system is so quick to react on these subsequent encounters that we fight this stuff off before symptoms start to arise, which is pretty amazing. But battling infections, like actually doing that fight, having to suffer that infection, is only one way to gain immunity. So let's talk about what our options are. The first one is the one that we already mentioned, which is what we call natural immunity. This falls into a type of immunity we call active immunity, which really involves getting direct exposure to something, to some kind of pathogen or the signature of some kind of pathogen, and mounting a successful response. It involves you, your body, making these antibodies and figuring out the recipe to do that and to fight these things off yourself. And so that's really what active immunity is. Natural immunity is when you do that by getting exposed to and contracting that pathogen and you develop that disease. This full mounted response usually occurs after the first, roughly 10 to 12 days. And it peaks when you have, this massive antibody production. You have your B and T cells are very active and they're out there fighting for you and things like that. And so once you have that experience, those cells that produce those antibodies that went out and targeted those individual pathogens, they are going to reduce in number, but they will always, for a long time, stick around in your body at low numbers and sort of be on call when they're needed, like when another infection arises, and that infection looks really similar to the one that you just had. So these cells are called upon and kind of brought back out to defend your body all over again. So that's a really cool thing that we have that ability to fight off these infections and then hold on to that memory for next time. We gain immunity that way, hold on to it, and keep it for if we ever encounter this thing again. But it's really important to note there are serious cons to this method of gaining immunity. There's an inherent risk involved in this because if you contract a disease, you will get sick. If you get sick, you can contract an injury. You can take an injury from that. It causes harm to the body, and in the worst case scenario, it can cause death. That it's not unheard of for a lot of these major diseases that have plagued humanity for eons to kill you. So it's really important to remember that natural immunity has serious problems. It's generally preferred that you don't get it. You don't have to deal with this bacteria or virus or whatever it is that we're talking about, and instead avoid it to the best of our ability. And even if we don't die from that infection, if we survive and recover from it, it could inflict some pretty long-lasting after effects that can come with that infection. More recently, we talk a lot about COVID infections because that's what is a big part of our life today. And COVID infections can result in the thing that we call long COVID. It's the result of damage that the virus has done to our bodies and to different parts of our body, our lungs, our brains, and it can leave people with long-lasting injury. It changes the way they live their lives. A more historical example is polio over here, where polio, not in all patients, but in some, would cause partial or widespread paralysis. And so some people who were paralyzed by polio were paralyzed at a certain point in their body that prevented them from breathing healthily on their own. And so doctors had to create a piece of technology like this. We call this the iron lung. And it was a device that would essentially, it would breathe for a person. It would create like a negative pressure on their body that would expand and contract their lungs for them. And so this, people that contracted this kind of paralysis became bound to these machines. They were unable to go about their lives. They were, essentially held captive in this thing. And so we obviously, those of us who are going down this health path, we want to be healthcare providers. We want to not just prevent people from dying of disease, but also reduce harm in every way, and prevent people from taking these lifelong injuries when possible. And it's also worth noting, when you have this disease, you are contagious with that disease. You can spread it while you have it to other people. So even if you are fine, even if you contract this disease and you don't feel anything, maybe you have a small headache or something like that, it's like, oh, it's fine, whatever, and you go out to go about your day, you are still, while you are fighting off this disease, you are spreading it. You are shedding it. It is going out into your environment and can potentially infect more people this way. Okay, and so it's really important to note. I still remember. I'm still quite bitter about a friend of mine that never still went to a party that, knowing that they had COVID, decided to go anyway because they were feeling fine. And, it's incredibly important to remember. It's easy to forget that when you have a disease and you are asymptomatic, you still have that disease and can very easily spread it to others. Okay? And so depending on how long that disease lasts in you and the different methods of transmission, you could become an agent of infection for this virus or bacteria, whatever it is we're talking about, and increase what we call the R naught of that disease. The capacity for like every single person that's infected with them, how many new people are going to be infected while that person is infected. And so essentially you're contributing to that problem if you get this disease, whatever that disease may be. So this can be particularly scary for infants and young children who are, when they're born, their immune systems are inexperienced. They haven't been exposed to the things that we have over the many years of our lives. We have gotten, although we may not even really remember it, many, many, many diseases, many, many infections that our body has had to fight off. Some of those things were more serious than others. And so, when people are born, they're born with a very limited immune system. And that immune system builds up over time. And there are some ways that we can increase the immunity of these of these children and of people that also have reduced immunity passively. We can do that even from infancy. That we can give people exposure to the things that fight these infections without having to give them direct exposure to the infections themselves. So, for example, when a baby is drinking breast milk, the breast milk contains antibodies that are produced by the mother that can be absorbed through the baby's stomach. This is actually something that it's not just for babies. Anyone can take medication orally that contains antibodies and absorb it that way. Antibodies are actually really hardy and they can resist the acid inside of a stomach, and so you can take them up that way. It's really interesting. And then another way you can kind of give somebody antibodies is through a blood transfusion or through otherwise like injecting this into a person's bloodstream. So because our antibodies circulate in our blood, we can take collections of our blood. We can take a donation and give it to another person who does not have those antibodies, and they can at least temporarily acquire those antibodies. And antibodies, they're pretty cool. They actually circulate in your body for quite some time before they eventually are removed. And so those previous ones were ways to gain immunity in some rather interesting ways. And natural immunity was a very direct method of exposure. These other antibody focused approaches are indirect in the sense that they are there's no exposure to the infectious agent, and instead you're getting exposure to you're getting like a supply of these antibodies. But you can also gain immunity indirectly just by being in a population of immune people. This concept is called herd immunity. And this, what this essentially means is that in a population, in a herd of organisms, of like a species, the immunity of other individuals can act as a shield to protect individuals who are not immune. And there will always be individuals in a population that are not immune to an infection. Even to the best of our ability trying to increase immunity, it will never, ever be 100% because, again, individuals are always being born in this population and we're not born with innate immunity. And also, immunity wanes for various reasons. People who have reduced immunity, who are immunosuppressed, they can have weakened immune systems, and so they can be put at risk of getting these infections, even if they've had the infection before. And so herd immunity is really important because these individuals, if they exist in a herd, a population that has a really high percentage of immunity, they are much, much, much less likely to encounter and get exposed to that infection because they're much less likely to get exposed to another vulnerable person, someone who is sick with this infection, who actually has it at that time. Herd immunity is an amazing thing. This is incidentally why we do a lot of vaccinations. We try to increase herd immunity to protect other people who cannot get vaccines. But it really depends on the percentage of the population that is immune. If the percentage of that population is too low, then it's still quite likely that a vulnerable person is going to get exposed to another infected person. And what that percentage is, what that target percentage is that we're looking for, varies a lot by a lot of different factors. The rates of transmission, the rates of spreading this infection from host to host, the R0 value, again, that is the for every individual who is infected with this virus, it's the average number of new cases that comes from that original infection. So for some diseases that can be quite high. For diseases like measles, measles is one of the most infectious diseases that we know of. That R naught is something like 12 or 16. It's somewhere in that vicinity. What that means is for every single person, for every new case of measles, that person with measles will go on to infect another 12 to 16 people. And so there's a lot that kind of goes into, what the effective percentage to achieve for herd immunity is. For a disease like measles, that percentage is going to have to be much, much higher than other diseases that are less infectious. Importantly, immune memory, it's not going to last forever. There are lots of reasons why immunity is going to wane over time, and it can also vary depending on the disease that we're talking about. That some diseases, after you get immunity, will last longer than others. A good example of that is measles, which lasts a long time. People believe that it lasts a lifetime, but that is not entirely accurate. It does tend to wane over the course of a person's life. For example, recently my dad went and got his immunity checked, and he found that even though he was vaccinated for measles when he was a child, he is no longer immune, and so he had to go and get another booster today, or not today, but pretty recently. And so this immunity doesn't last forever. It'll eventually fade, and for some things it fades faster than others. COVID is another good example. It famously, it's pretty easy to get reinfected with COVID. So we get immunity that lasts, a few months to a few years, and we still don't really quite understand the amount of time that it really lasts. And then, of course, we have other situations like becoming immunosuppressed, that some patients that have autoimmune diseases or suffer from cancer and are getting treatment for cancer, or have HIV or other diseases that otherwise attack the immune system, this can cause your immune memory to fade. It can make these records, these experiences of past infections go away, which makes you now vulnerable to all these old things all over again. Today we can actually, like I said before, we can check a patient's capacity for immune response with a lot of new science technology. We can do something called titer testing. And this is when we look for a quantity of a specific antibody that targets a specific disease in the blood. We're pretty good at figuring out how cells are going to create antibodies to attack a specific feature of a pathogen. So we can look for those specific qualities in an antibody in your blood and determine, is this present? Is this present in high numbers? Is it present in low numbers? Is it not present at all? And if that number is too low, this indicates that immunity is waning and it's starting to go away. Now, ideally, what would be great is if we could give somebody immunity to a disease without having to actually contract the disease. This would be really ideal because what we're really hoping for is a significantly reduced or very minor exposure to these diseases, or maybe not even exposing them at all to it, and instead we expose them to like a killed pathogen that can't cause disease, but our immune system could still attack and use as a source of experience. Of course, we can do this because that's what a vaccine is. So what we're talking about here is what we call artificial immunity, another type of active immunity, much like natural immunity is. And so artificial immunity is any kind of exposure that you gain to particles, infectious particles, or infectious organisms associated with an infection, but that in a way that does not cause disease. If this is a significantly safer exposure that does not result in the manifestation of the disease as we know it, and instead is an easy way for our body to, find something that looks like a disease, attack it, create that immune memory, and gain experience for fighting this disease off without ever having to actually fight it for real. This way, if we ever encounter it again, we have that very targeted mounted defense, and we can quickly mount a response. It's worth noting today that as I'm recording this, there's currently a pretty substantial measles outbreak that is going on, particularly down in Texas, but is also spreading to other states. It's become national at this point. Measles, again, highly infectious, highly infectious, very, very high R0 value, and the cases, by and large, like predominantly are occurring. Like 90+% of these cases are occurring with patients that have never been vaccinated for this for measles. A very small percentage of people that have been vaccinated for measles have contracted measles during this outbreak. But again, it's worth noting that immunity doesn't last forever. So it's possible in some cases that immunity waned enough to make that person vulnerable again. But the key here is that, remember, measles, so infectious, extremely infectious. And there are more people in America who are vaccinated for measles than those that are not. Yet, there is still a staggering slant towards the cases of measles being almost exclusively, except for a few cases, people that have not been vaccinated for measles. So this is a very effective defense against these diseases. So artificial immunizations are a type of prophylactic medication. Okay, this is a preventative treatment for a disease. It's that we don't have this disease, but we're going to take this medication to make sure that we keep ourselves from getting it. There are lots of different kinds of prophylactics that we take throughout our lives. Some people will take birth control pills as a prophylactic for pregnancy. People will go get dental cleanings as a prophylactic for cavities. People will put Neosporin on cuts as a prophylactic for a skin infection. So there are lots of different kinds of prophylactics. This is just one type that we take oftentimes very early in our lives and throughout our lives to make sure that we don't actually contract these miserable diseases. I'm going to talk about sort of the start of all of this immunization because it's kind of a cool story. So there's a lot more information about this. I'm going to keep it pretty short, to be honest, all things considered. But artificial immunity is actually a really old kind of treatment. We have been doing something akin to a vaccination for millennia, really. We have writings that emerged from India and China in thousands of years ago, that a few thousand years ago, that show methods of what we call variolation. And this was a strategy to fight a really, really problematic disease that doesn't exist anymore because of vaccination, called smallpox. Variolation of smallpox kind of went like this. So first, smallpox, let me just say, a miserable disease. It was horrible. I don't know if I have any pictures in this slideshow that really represent this, but when people would get smallpox, they would develop horrible, painful sores all over their body. They'd be racked with pain. It would scar you for life with these wounds. They would turn into pox that would mar your face, your arms, your legs, your body, everything, and they would stay with you for life. And it certainly could kill you as well. It's really worth noting that there was a large death toll that was associated with smallpox. I believe it was something like 30% of patients that contracted smallpox would die of it. So it was really impactful. Essentially, what variolation was, it was a method to expose somebody to smallpox, but in a very controlled way to minimize the potential for them to experience symptoms and especially to die. So the method went like this. Essentially, you would take a patient that had smallpox and you would extract fresh pus, right? Very exciting, very, not gross at all. Extract fresh pus from one of these sores on the patient. And it was really important that the patient's case, when they went to look for a patient that was a suitable sort of donor, it had a mild case, because it was an indicator that this, whatever was going on here, it was not so bad. So it was a little bit easier. It was more likely that if you expose somebody else to this case of smallpox, it would also not be so bad for them. People didn't realize at the time that they were not just collecting pus; they were also collecting antibodies, active antibodies that they could add to a person's body and kind of kickstart this process. And so then they would take this pus, they would allow it to dry, and then they would grind it up into a fine powder. And then they would administer the powder in various ways. Initially, some of the things that I've read involves blowing the powder up a person's nose, but that's a pretty old approach. Up until variolation fell out of favor, it was instead scratched onto the skin. So a doctor would take a knife. This is Edward Jenner, very famous person in vaccination. We'll talk about him later. But these people would administer the powder by scraping the skin with a knife, and then rubbing the powder into the wound. Okay, not a fun experience by any means. The next step would be to wait for the patient to develop symptoms of the infection, because don't forget, with variolation, you are giving a patient this infection, but it is a very controlled infection and is ideally a less intense and significant form of it. So variolation was actually quite successful, and it was done for, again, thousands of years because of two reasons. These are why it was so successful. First, when you would expose a patient to variolation, it was in a very, very small dose. You would add only a few viral particles, really. Like it was pretty reduced, because you're grinding up a single scab, and which has a lot of immune activity is already happening here. So there's antibodies that are there. There's lots of white blood cells that are there, and they're actively killing a lot of these viruses and things like that. From a mild case, the viral load, the amount of virus that they had in their body, was low to start. And so it was really important. And so the second part is port of entry, which is essentially the path of infection, the route that an infection takes to infect a person. That was very important. What we had learned was that the disease spread best through the respiratory system. If you breathed it in through your nose or your mouth, you really increase the capacity for that virus to spread throughout your body and cause lifelong, body-wide problems. That would be lifelong in the end. But it would spread quite aggressively if it entered your body that way. But we learned that if it was introduced to a person through the skin, the ability for it to reproduce and to spread throughout our body was much slower, and it was also much more localized, which meant that our immune system could kind of get an edge, start actually fighting this stuff off, and it was easier to control when it was introduced this way, which is again why the skin scraping method became the main method that that variolation was applied. Now, variolation was not without issues. There were a bunch of different kinds of problems. Still, there was a notable mortality rate that about 2 to 3% of people would die of this infection that they got there, because it's still a deadly virus. It was very problematic. And so even a mild case could kill a person, especially if they had other issues going on at the time. People had plenty of health issues, especially back then. Still, 2 to 3% was definitely better than 30%. So this was a marked improvement that protected people as a whole. And people still developed symptoms of smallpox during this time. So you get exposure. A mild case of smallpox was still nothing to scoff at. You'd have massive, blisters and these sores that would develop on the part of your body that the infection was administered to. So if it was on your arm, if it was on your left arm, well, your left arm would be kind of encrusted in a lot of places with these wounds, and that they too would scar after they were, fully recovered. So they would leave long-lasting wounds on people. And also, variolated people could still spread smallpox. That was still an active infection. So while a person was undergoing treatment, they could transmit it to others. So variolation was helpful to keep people broadly from suffering from smallpox, but it was not an effective tool to necessarily keep it from spreading when people were infected. It could still go out and about. A person who was suffering from who had gotten variolation could still shed viruses, and if other people who were not variolated or had never had smallpox before got exposed, they could still contract it. So still a lot of problems. There are more too, by the way. There's a serious issue of accidentally spreading other diseases while doing variolation, because of course you would take tissue, blood, pus, things like that from another person who could have some other disease, like, I don't know, syphilis, and essentially inject that, scrape it into the skin of an otherwise healthy person. And now that person, yes, they have smallpox for a short time, but now they have syphilis. So there was definitely many additional issues that came with variolation. It was not a perfect system, but it was what people needed at the time. Enter Edward Jenner. Okay, so Edward Jenner is this English doctor, scientist, naturalist. These people that were like polymaths. They do everything. And this is a guy who's responsible for the first vaccinations. At the time, in the 1700s, it was a known phenomenon that dairymaids, people that would work with cows and milk them to make cheese, to make yogurt, to make milk, that they were very unlikely compared to the broader population to contract smallpox. And this was because they often acquired cowpox first, a disease that is very similar, very genetically closely related to smallpox, that was pretty exclusive to cattle. But it could occasionally jump from cattle to people, and it would cause similar infections. So these pictures down here are actually a much more modern case of cowpox. People still get cowpox to this day, and it causes a lot of, it's not comfortable, it's painful, but it's a very mild disease compared to smallpox. So what Edward Jenner noticed was that he kind of put the pieces together, right? This idea here, dairymaids were unlikely to get smallpox, that was well noted at the time, but it wasn't clear why. Edward Jenner put the pieces together and realized it's probably because they're contracting cowpox, which seems very similar to smallpox. And so is it possible that if you contracted cowpox, which was much more tolerable, much less likely to result in grievous wounds, scarring, death, if you contracted cowpox, could you get protection from smallpox? And so Jenner, he designed a method to do this. He essentially used the same approach from variolation but he took pus from a wound from a person that had cowpox instead, and he introduced that to a person. And what he found was that first the resulting recovery when you were vaccinated for cowpox, it was very minor by comparison. Here's a good illustration of like what it looked like when a person was variolated for smallpox. You'd have these massive blisters that would expand all over the arm from the site of the scraping. But if you were instead vaccinated with cowpox, it was a really different experience. Your body reacted much better to it. It did not cause such significant injury to you, and it was certainly less likely to cause death. And Jenner was right, by the way. He was right that getting initial exposure to cowpox did in fact protect you against smallpox if you ever encountered it later, because those viruses were so similar and our defenses against them were so similar that they were able to attack. If you had the defenses against one, they were able to work against the other. So Jenner used vaccination as the term to describe his new method, where it's a reference to cows, vaca meaning cow. So vaccination is this process about exposing someone to cowpox rather than smallpox. And since then, we've kind of taken that term and used it for all kinds of stuff. So everything is now a vaccination. All of these types of artificial exposures become vaccinations. And there are lots of different kinds, by the way. I'm going to talk about a handful of these here and then show some more. So we have the live attenuated, otherwise called the weakened or inactivated vaccine. This is where you have a whole virus, usually virus, but could also rarely be a bacterium, that is alive, or as alive as a virus can be, so not living but functional. And then we have, and it's adapted, it's changed in the laboratory. It's made in such a way that makes it very difficult for it to infect and cause disease in a person. We often do that by having the virus infect chicken eggs, and then have many generations of these viruses infecting chicken eggs over and over and over and over and over again. And eventually the population of this virus evolves to become more specialized to parasitize chickens than it does to parasitize and infect humans. And it can change so much. Evolution changes these viruses so much that when you then turn around and expose a human to them, this new modified version of the virus, it doesn't stand a chance against our defenses. Our defenses, really take over and destroy this thing really quickly before it can cause any significant disease. As opposed to that, we can have a whole killed organism, which can be, again, bacterial or viral, usually viral. And this is where we take the virus, or the pathogen in general, and we kill it, usually with heat, sometimes with chemicals, the chemical sterilizers, and then we add that to the vaccine and inject the person with that. And so here we have a pathogen that is no longer functional. It cannot cause disease, but it's an intruder, or recognized as an intruder in our body, and so our immune system still attacks it as if it's foreign, as if it's something that's causing disease. We still get those immune responses, and we create these defenses that have our body ready to intercept that virus if it ever shows up again. Some vaccines are toxoid viruses, which are really different. These are when we have like a modified product for a disease. oftentimes a bacterial disease like diphtheria and tetanus, that these bacteria produce toxins that cause most of the symptoms that lead to severe injury and death for people. And a toxoid vaccine is when we take that toxin, we modify it a little bit, we make it non-functional and look very specific to our body, and our body can target it, create antibodies against it, and attack it just like it would a virus or some other kind of pathogen. And so this really helps us quickly fight off these infections, because we don't have to suffer the significant impact of those vaccines. Then we have the subunit vaccines, right? These are when we have pieces of a virus or a bacterium, usually like a purified protein or pieces of a protein that we have smashed together, that are all parts of the same pathogen. Again, these are distinct, separate chunks of a pathogen that the thing is dead now. It cannot cause disease, and you inject that into a patient, and as a result, it creates in them an immune response. Okay. We also have other things we can create. We can create like fake viruses. These are virus-like particles that we essentially make like lipid membrane, and then make pieces of the virus that we stick to the outside of them. These other things I'm not going to have you remember too much about. Really, though, I want you to remember these four especially, and one we're going to talk about later. We also, we can do a lot of crazy things now, where we can create some very, very unique vaccine approaches that let us essentially create fake viruses or modify harmless bacteria to carry the things that we care about, being recognized, things like that. And we take these vaccines and we administer them through some pretty different routes. Some of these methods are more effective than others. Some of them are preferred more than others, and they also can depend a lot on the type of vaccine that we're talking about. Again, as we saw with variolation, the route of administration really matters. There are some vaccines that you should not administer one way, you should only administer the other. Some vaccines are more flexible. So here are some different ways we can administer. There are some vaccines that can be administered orally, as liquid drops in the mouth, or as potentially like a pill, which talks about that kind of thing, which is pretty cool. You can have an intranasal spray, which is where you inhale through the nostril. Something like swine flu historically has been vaccinated this way. I remember that I got that kind of vaccine way back when. But the vaccines that we're more familiar with are administered one of two ways usually, and that is the subcutaneous route, which is being injected under the skin and in fatty tissue. So that's like the dermis or the subcutaneous layer beneath it, and then we have the intramuscular route. So this is when we're just injecting directly into the muscle. And so these different sites, like where we do the injection and also which one of these we do, can vary a lot depending on a patient's age, their height and weight, and things like that. So it definitely can vary. So vaccine types. They don't just differ by the method of immunization, but they also have their own unique pros and cons. So some methods allow the pathogen to actually reproduce in the host for a limited amount of time, which sounds scary. It does sound scary, but these are very controlled exposures, remember. So the benefit of this is that these types of of vaccinations, the ones that allow pathogen replication, end up inducing a much more significant immune response. So the result is like your immune response is very strong. You're going to have a lot of antibodies produced, and it typically associates with a longer memory as well. That these immune memories, the immune responses can be stored for longer. So the let me see. Then we have other methods that do not allow further propagation. So these are methods that have a very capped immune response. These are dead cells, mostly dead pathogens, things like that. Okay. Then we have the mRNA vaccines. Okay. So this is when we have a vector particle that this is essentially a molecule that is not the virus. It's separate. It's just an agent to carry a piece of mRNA. Okay. And the cells, what they do is they take up this mRNA, which codes for a single piece of the virus, not the whole virus, and it's taken up by the cell and used to make a fragment of that virus, a fragment that can then be the target for an immune response. Okay. So this is a really new technology. We saw this most recently with COVID-19, and people are starting to use this for a lot of new stuff, which is very exciting. So a lot of pros to this new method. Now, there have been some major changes to the world post-vaccination. Vaccines kind of came onto the field pretty late, all things considered, for humanity. A very recent thing that really came about in like the 1900s. Proper vaccines as we know them today, and they really changed the game. Where now a lot of mortality from infectious disease has dropped off dramatically. Where people are no longer dying of these very preventable diseases anymore. Still in some places that still need a lot of attention, but generally speaking, this is a huge improvement. I'm going to end this by talking about some common misconceptions about vaccines. Okay, some people say a lot of these things, and I'm going to kind of address them because they're very important today. Some people say, I'm a healthy person and I have a strong immune system. I don't need to take this vaccine or the other. I don't need to get my annual flu vaccine. I don't need to get my COVID vaccine, things like that. Some people say that natural immunity is always better than vaccine-induced immunity. Some people say that vaccines like mRNA or rDNA vaccines, another type of vaccine, that they are capable of rewriting your DNA and changing who you are as a person. Other people say that, healthy and careful living or like specific diet or things like that, drinking raw milk, I don't know, weird things like that, are replacements for vaccines. Well, here are some of the answers to those issues. Being a healthy person, for one, does not make you immune to an infection. And a vaccination severely reduces the risk of passing that infection on to others as well. So if you get vaccines, you are not just protecting yourself, but you're protecting other people around you. Natural immunity is not better than vaccine immunity. Natural immunity has some serious issues with it that can cause injury, death, and again, transmission of that disease. It's impossible for an mRNA or rDNA vaccine to rewrite your DNA. That's just not how it works. There's no relationship between those things. There's no crossover between your genome and these pieces of DNA or RNA. There is none. And also, healthy living, diet, and exercise only goes so far. They are not going to fight a pathogen, let alone prevent it. It can help us along that way, but it is not going to keep you from getting measles if you are exposed to measles. So, with that, that's all we're going to talk about today. So hopefully that also provides a lot of background information, a lot of maybe some guidance for potentially how to talk to some family or friends that have these misconceptions. As healthcare providers, it's pretty important that we serve that role in our community, that we recognize the importance of vaccines, getting our vaccines, helping other people get vaccines, and take care of themselves. So, with that, I'm going to end today, and I'll see you next time.